This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, a special episode recorded on August 17th, 2022. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Today, I am in Ithaca, New York at Cornell University, a place I spent 4 years of my life as an undergraduate from 1970 to 74. I'm back and I'm doing a series of podcasts, and this one is with a professor in the Department of Molecular Biology and Genetics, Cedric Feschotte. Welcome to TWiV.
Thank you, Vincent.
We actually did some of your work on TWiV with Nell Zeldy years ago.
Yeah.
And we always wanted to have you on that podcast. But I thought since I was here, I would stop in. So thanks for spending the time and talking about your research.
Coming back to your roots.
Yeah, my
graduate roots.
But you know, when I was here, I had no idea I would be a scientist. I didn't know what I wanted to do.
So what major were you?
I was a biology major.
Yeah.
But I graduated with no plans. So
Me too.
I figured it out. But this is about you. So tell me your history. You're from France.
I'm from the southwest of France.
Which city?
Toulouse.
Toulouse. Okay.
Yeah. This is where I was born. This is where I grew up. This is where I did my undergraduate studies. Mm-hmm. Biology too. But actually my passion at the time was focused on bugs, insects. I wanted to study insect behavior. I was really interested in ants, social insects.
Right.
Because there was a group studying these, but I wasn't really sure that I wanted to do research in academia or any other settings. I just was not thinking also very deeply about this. But I was a naturalist. Really, that's what drove me into what we do now. Actually, is looking at creatures out in nature. Thinking about them, collected insects.
So this is just a childhood interest, insects, right?
Yeah, insects. One day was insects, another day would be birds or snakes, the animal world. I really like this. So anyway, Toulouse. And then because the year I was actually graduating, my plan was to stay in Toulouse. Because, French, tend not to travel as much and move around as much as Americans would. So I thought I would stay in Toulouse and go on a PhD program in behavior. But the year where I was planning on applying, the program actually didn't open that year. I think they ran out of funding or something like that. And so I was like, well, what am I going to do now? That's my only plan, So what's plan B? Well, anyway, I did apply to other graduate programs elsewhere. And I got into one in Paris, and this was called insect physiology. And I really was not very well prepared for what I was gonna do. It was very sketchy. I made it barely by an inch in the program, and then they're like, "What are you gonna do?" And I had a bit of a plan. They're like, "No, this doesn't make any sense. What else?" And they give me a piece of paper that we had, a title of a potential graduate project with different labs, not just in Paris, but all over France, because in fact you could, join different labs that were affiliated with the program. Anyway, one of them the title was The Ecology of Mosquito Transposable Elements or Mobile Genetic Elements, or something like that. The ecology of the genome, or something like that. It was like ecology of the genome, mobile elements. That sounds very intriguing. I had no idea what it meant because I had very little background in molecular biology and genetics. This was going to be this program was heavily molecular, and was going to be my entry in a way into this world of molecular biology and genetics. So really, honestly, I just didn't really know what it meant, but it sounded cool. And it was the research lab was located in the southwest of France, again, a bit closer to home than in Paris, where I did the coursework. Then you go do your research in this lab in Pau, University of Pau, PAU, which is a small university in southwest of France. And I met this Professor Claude Mouchès, who was studying mosquitoes and genomes and transposable elements. So this is how I got into it.
By accident.
Kind of, yeah. It really, actually, really is. I mean, based on a couple of keywords, you know. Also, I met him when he came and gave a lecture in the program in Paris, and I met him, and he was very passionate. He was actually trained as a virologist, it turns out, and studying plant viruses in France, and then he switched to like study mosquitoes and insecticide resistance, and discovered actually at the time a new mechanism of insecticide resistance, which was amplification of esterase genes. So these are they encode detoxifying enzymes, and the resistant mosquitoes had 250 copies of the gene versus only one copy in the sensitive mosquitoes. And it was kind of a big deal. It was a science paper in 1986. And when they started sequencing the amplicon that was amplified in these mosquitoes, these resistant mosquitoes, they realized that the amplicon, instead of just the esterase gene, which is relatively small, was a much larger piece of DNA. And of course, they started to sequence laboriously. This is in the late 80s. The whole thing, they realized it was stuffed with transposable elements of some sort, retrotransposons. And they were actually some of the first, maybe the first described in mosquito genome. So then he switched to these transposable elements. And the big idea, which just goes back to like McClintock now, is the genomic shock idea, where maybe in condition of stress, under, for example, pesticide stress or, treatment, maybe mosquitoes are able to kind of remodel their genome through these elements. And that's why they use some mechanism to amplify this esterase gene through the activation of those elements. So anyway, long story, but I joined the lab to test this idea specifically. But we had no active transposable elements discovered in mosquitoes, so those that are actually capable of jumping, and we'll talk more about this, but this is a relatively small fraction of any genome. So my goal as a graduate student was to hunt for those.
In mosquitoes.
In mosquitoes, but in the absence of genome sequences or anything like this, right? So phage libraries and and I'm not going to go into the details, but...
So we have Corn here from Barbara McClintock. So was she the first to imply mobile elements based on genetic data, not sequencing at all, right?
Oh, yeah. Cytogenetics. So broken chromosomes is what she was tracking then. This is in the 40s. And realized that a chromosome in particular was breakable at a very specific site, which she called dissociation. And realized that the breakage was a transposition reaction, but it was actually an aberrant one that led to the loss of the chromosome. So very dramatic chromosomal event, so she could see this under the microscope and track it. And then she realized that this breakage only occurs when you bring in another locus, which she called activator. And that became the ACDS system of Maze, which was the first system of transposable discovered. And it basically laid down the groundwork, the foundation really for a lot of the things we know about transposable elements, this kind of activation of elements in trans and so on. So but the genomic shock, that was an idea much later on, that these elements were sort of integral part of the organism, that they were being used to regulate gene expression. For one, this was really the thing that she was really interested in development. And also in condition of stress and in response to the environment, the genome could remodel itself. Gene expression could change dramatically through the mobility of these elements, and they're spreading throughout the genome. So this was our big ideas. Those were not the ideas, by the way, which were widely accepted at the time. The mobility of DNA was, in fact, this is what led to receiving the Nobel Prize in 83, I think, 83. But that was really not the endpoint for Barbara McClintock. She really wanted to understand how these elements, affect the biology of the organisms and development in particular, plant development. Yeah. We've moved away a lot. But I can go back because, in fact...
I think the history is of course, so she was really the first. And then as we moved into the molecular era, we could start to see them in the DNA, right?
Yes.
We started to be able to sequence genomes.
Yeah, so that's the first sort of major, I think, transition in the study of transposable elements, is when we move from genetics to genomics. Right. Of course, we moved to... there's a molecular characterization of the element in the early 80s. That's a very crucial step, obviously, as well. But at the time, in the 80s, early 90s even, people were studying active transposon elements because they were studying the mutations that these elements cause that were visible in flies, the white eyes, the corn, the kernels, and other phenotypes in bacteria, antibiotic resistance. They was based on phenotype and what the elements do when they hop. Right. And then people started sequencing genomes, like in an unbiased fashion, right? What's in the genome? And it was very quickly realized, it was already known that genomes were chock full of transposon elements or their remnants. We can go back to this, but this was already inferred from reassociation kinetic studies of Roy Britton and other people. But anyway, so when genomes getting sequenced, the shock was like, well, yes, indeed, like the human genome, for instance, so pretty much most of the large eukaryotic genomes were replete with those elements, but most were not clearly capable of transposition anymore. So that really triggered a big shift in how we study the elements. Instead of studying active transport elements, now we study them as a population, as a whole kind of ecosystem. It's kind of where I started my work at the transition between the sort of molecular characterization of and hunting for active mobile elements to focusing on dead elements in a sense. Dead in terms of their mobility, but looking at their impact nonetheless on the evolution and the function of genomes through other ways than just jumping. Right. Because again, it was very clear that even in maize, even for McClintock's work, even in fruit flies, where there's a lot of activity, mobility of the elements, but still it's relatively rare, right? So that's a big shift, genetics to genomics. And then there is another big shift, I think, which is a modern functional characterization of genomes, where now we can map the activity of genes and the chromatin genome-wide, a bit more or less unbiased, and we can talk more about this, but through, next generation sequencing type approaches. And the big shift here is when, sequencing reads start to get a little longer. So for us, this was a major turning point in not too long ago, but maybe 10 years ago now, something like that. Reads starting going from 30 nucleotide to 100 nucleotide. And that makes a huge difference in the ability to map those reads onto the genome and to see into this kind of dark matter of the genome, the repetitive fraction of the genome, which is what interests us. This repetitive element, these transposon elements. So now, for the first time, we don't just have the sequence of the elements. We also have an ability to look at their activities, their regulatory activities, their coding activities, right, their expression, in a more or less, again, unbiased fashion, less and less biased, so a more holistic way, And I think that continues to this day now. Long reads, again, facilitate the study of those elements. So I think we're going to enter yet another new phase where we're going to see even more. And I think this is where it's drawing more and more research, because I often wonder, where is it that not more people are studying these elements? Since, after all, it should be 50% of people working on the human genome. It's much less than that, right? Yeah. We all focus on 1% of the genome.
The protein coding part, right?
Right. So why? Well, and I think the answer I would have is twofold. First, there were dogmatic reasons, meaning, in the early 80s, there's two very important papers that were published about putting forward the notion of selfish DNA. Orgel and Crick, Francis Crick was one of the papers, Doolittle. But that was sort of came with a bit of pejorative kind of view. And they certainly didn't say selfish DNA or parasitic DNA. Doesn't mean it's not interesting. But people were well, probably not that important because we can explain the presence and the spread of these elements without inferring that, without evoking that they have a function, So that kind of I think maybe, I don't know, I'm just speculating that it might have discouraged some people to work on this part of the genome. But then I think mostly it was technical. Technical because early on, again, everything was much easier with your single copy gene, essentially. Removes a lot of the technical hurdles that you have And that's true to this day when you deal with this mass of elements. And again, it's from shifting from like studying one element to try to study all of them in the genome. Right.
I think another reason is probably funding. People found it difficult. I mean, because initially you have to do some kind of expedition, right, and explore, and then funding agencies don't like that. They want you to have a hypothesis. Right?
That's right. Yeah, it's initially when you mine, explore what's going on, you really, and indeed, if you say, well, I'm actually not even looking at elements that are moving around, and you can see how this would be.
So you were introduced to these elements in this mosquito lab where you did your PhD, right?
Yeah.
And then did you do a postdoc after that?
Yeah. So then, the thing is, like, I really underwent this transition of like being molecular or trying to be a molecular biologist at the bench, which again was a challenge for me, to like starting digging at the computer, searching, sifting through genome sequences. So we're talking about the late 90s, early 2000, right? And this is really, there's a massive, expansion in, genome sequences available in databases, and you can do blast, homology-based searches at the computer was like my weapon of choice. I would just do this all night. Look for stuff with blast by homology, taking a mosquito element. And so, in fact, my first paper is not on mosquitoes, it's on Arabidopsis in the plant genome. And again, the story is real. It's like starting from a little bit of sequence I had in mosquito, I ended up in the Arabidopsis genome chasing these related elements and finding some interesting things about them that was not known. So then that I realized this is very important, because I realize now there's no boundaries. We can actually, especially if we start our work at the computer, we can really explore any organism we want. And all of them virtually are stuffed with elements. So this become very exciting for me, because I'm like, first, I can go anywhere I want. But I have questions, right? It's driven by the biological questions. And at the time, I stumbled on this class of transposable elements. They were called mites, miniature inverted repeat transposable elements. They were discovered in plants by a person who became my postdoc advisor, Sue Wessler, at the University of Georgia, then discovered that these elements were very abundant in the maize genome, in the rice genome, in grass genomes that we're looking at. And they were very mysterious because their copy number didn't suggest some sort of replicative mechanism of transposition, yet they looked like the cut and paste element that McClintock had found. So, which generally are low copy number because they excise and reinsert in the genome, so they don't have really an amplification step, if you want.
So it's like a computer cut and paste. You remove it and put it somewhere else, as opposed to copy and paste, where you leave one and then you make another copy, so that amplifies it, right?
Yeah, and these are the two big classes of elements now, as we know them in their classification. Is the cut and paste element and the copy and paste. Those are the ones that go through an RNA intermediate that's reverse transcribed back to DNA and reinsert into the genome. I think we're going to talk all about those type of elements in a minute. But at the time, so my focus was on these other type of elements, what we thought were this special class of cut and paste elements that we call mites. But we didn't know how they moved because they were very short. There are no coding sequences, no transposase. So they were really mysterious. But with the availability of full genomes, we're like, well, we're searching the genome. We should find their partner, their master elements. So that's what I wanted to do. And I wanted to prove that indeed they were mobilized in trans, just like McClintock had described with AC and DS. And yet they find a way to amplify through mechanisms that we still don't completely understand, but we have some ideas. Anyway, so then I joined to do this. So that's very specific. So this time it's not at random. It's very driven because I had a very specific question, and I found that Sue Wessler was, obsessed with the same question. So then, that's great, right? You go to where you think is the best place, I thought was the best place in the world to kind of solve that particular problem. And then I kind of then switched, but seamlessly, really, because a lot of the work was computational anyway, seamlessly, I would say, from mosquitoes or to like the rice genome is what we studied at the time, because it was the very first large plant genome to be sequenced. After Arabidopsis was a small genome, rice was a grass. We know it's full of those type of elements, these mites. So I dug into this. So that's that. So I was a postdoc at the University of Georgia in Sue's lab for 4 years. And then I started my lab at University of Texas at Arlington in the Department of Biology, first faculty position. And I was there for 8 years. And then there I started, I went back to animal genomes. So I started focusing on vertebrate genomes, including the human genome. Mm-hmm. On those elements, again, the DNA transposons, the cut and paste elements. Because not many people were looking at those in the human genome. You know, a lot of people were interested in retrotransposons because they're much more abundant in the human genome. But there was very little known about those type of elements, the cut and paste ones. And yet, you know, they make about 3% of the human genome, so that's a lot of DNA. And then that's where we started working on, and that took us to bats, by the way. Because early on, we realized, well, those type of elements, those DNA type transposons have not been active in the human lineage for very long, which is in part why people were not so much looking at them. But then one day we had the bat genome, and we looked in the bat genome, and a bat genome, the little brown bat specifically, and the little brown bat genome was chock full of very recent DNA transposons, actually.
How do you know they're recent?
Yeah, so how do we know they're recent? So that's all about like looking at their sequences. So it's a very simple idea. Like, so as you know, these elements can be, there's not one or two types of elements in any given genome. There's lots of different types, and they make families of related sequences that have amplified from a master initial original progenitor element that seeds copies throughout the genome. So at the time where this copying is happening, all of these elements, if it happens really quickly, which it usually does, all of these elements sort of are identical to each other at the time, or very close to each other when they integrate in the genome. And if there is no selection acting to preserve their sequence, which generally there is not a lot of that, there are exceptions, but we can talk about them. Generally, we see that they neutrally decay at the rate of the clock of the species.
Right.
Like as non-functional DNA, if you want. And therefore, that means that the older they are, the more diverged from each other they are.
Right, okay.
And that's how we infer this.
So you can see if you have an array of repeats in there, similar, then they're recently duplicated.
This would be identical to each other if they copied yesterday.
Okay.
Yeah, and by the way, this was already inferred by reassociation kinetics, by Rob Britton and colleagues in the 60s. Mm-hmm. They wrote a paper in 1968 showing that human genome is composed in great fraction. I don't know remember if they put a number. Now we know it's about 50-60% of the genome of repetitive elements interspersed. They knew already they were not like 10 repeats. They were dispersed throughout the genome, and they look like they could be they could have transposed. They could have been transposable, but they already infer right away. But there was a great variation in the level of sequence divergence between these types, and they could infer there were different families based on the pattern of reassociation.
So this is all based on reannealing of DNA. Yeah. You take double-stranded DNA, you melt it, and then you can measure how fast it comes together. So if there are highly repetitive elements, they anneal very quickly.
And if they are close to each other, they reanneal so quickly.
And then if you have unique genes, it takes longer for them to reanneal because they have to find each other.
That's right.
And then you can calculate the fraction of each. I remember that as a graduate student, that was what we had. We had no cloning or sequencing. We had reassociation kinetics. You could do a lot with that.
And it's remarkable, if you read this paper, all the things they could infer about the human genome in terms of the families, the copy number of some of those elements, the very abundant ones, like we know now, Aloe elements, which then Rob Britton actually spent the rest of his career studying Aloes. In the 80s and 90s.
So when I was at Columbia, they had cloned Aloe elements on plasmids. So Aloe elements are rich in the restriction site ALU1.
That's right. Yeah.
And that occurs every 5,000 or so bases in the human genome. Is that right?
Yes, that's about right. It's one every 3 or 4 kb of DNA because there's about more than 1 million Aloes.
So let me tell you the experiment we did. We had taken human cells which can be infected with polio. We wanted to identify the poliovirus receptor. And we knew mouse cells did not have a receptor. So we took human DNA and put it into mouse cells. And we did that several times, and each time checking for the ability to infect with polio. So then after 3 or 4 transformations, we have mouse cells with human DNA. We want to pull out the human DNA. We use an Aloe probe to clone out, because the idea is there's an Aloe site close to most genes, and we pulled out the poliovirus receptor gene with a human Aloe. Isn't that cool? That's the way we did it. That's the way we did it back then.
Yeah, they're specific to primates.
Because we couldn't just sequence everything. We had to have a way to pull it out. Nowadays, you have all genomes, so you can just look at it and look what you want, right? But that's how we did it using Aloe. So I have a fond place in my heart for Aloe.
I need to go back to this. It's very interesting.
Nobody cares anymore. It's all
I'm actually kind of thinking about things like this right now in terms of genome editing, in the sense that you can take advantage the fact that these aloes are just so abundant, right? There's 1 million of them, and you're right. Every single gene is associated with multiple of those aloes. So think about it as a platform to multiplex genome editing. I think Joe Church has a paper about this. Okay, I'm not the first one to think about it, but it gives you an entry point, right? Because you make guide RNAs with CRISPR, you can target.
Right.
Essentially, I don't want to say 1 million copies. It depends how you design your guide RNAs, but a very large number of sites. And do this multiplex base editing is what they want to do. It's very interesting. Cool. So we might go back to these ideas.
So the thing, we had a southern blot for each stage of the mouse cells, the first, the second, and the third, right? And we probed the southern blot with an Aloe DNA probe. And the first cell line is a schmear. Less of a schmear. Then the third, you could see bands. Because we've enriched for the receptor. We've gotten rid of everything that doesn't confer susceptibility to infection. And then we could pull out those. We made libraries and bacteriophages and hybridized.
That's really cool.
It was a lovely project by my student, Kathy Mendelson, who...
This sounds like a lot of work.
Who, by the way, did a postdoc in France with the famous guy who used to work on hormones. I forgot his name. But anyway... Chambon, yes, Pierre Chambon, right. And now she's at Columbia. So in Texas, you got into mammalian genomes. And then after Texas, you went to Utah. Is that correct?
Yeah. And in Texas also is where I stayed in Texas for 8 years. Right. And this is also where we started getting interested in endogenous viruses. This was driven by also a new postdoc that joined the lab, Clément Gilbert at the time. And I don't know, I remember, how do we get into this? Oh yeah, I don't remember now. It's... this is when the first report of an endogenous lentivirus was reported in the rabbit by Rob Gifford and Aris Katsoulakis, who kind of pioneered this idea of paleovirology, looking at these fossils. And I love that as a naturalist. You tell me fossils, and I like it.
I remember that. Oh, I remember that. The rabbit. Yes.
The first, so a virus that looks like HIV, the same group, which no one had ever seen integrated in any genome, including the human genome, not in the germline genome,
right?
But they had found some relics of them, called relic, in the rabbit genome. And I was like, oh, that's really interesting, this idea that you can mine these ancient viruses and have access to, and they make the case they explain it very, very nicely. It's like, you have these HIV-like viruses that are millions of years old. And so you have access to deep viral evolution at a scale that's just very different from what they do when they study an exogenous infectious virus. You can not go back in time very much. Your coalescent time is very short because they evolve very rapidly. So that was fascinating to us, and we were like, okay, let's just start mining also genomes for things like this. And it was a bit of a race at the time to find different first endogenous virus of this kind and this kind. We competed with the same group and Clément actually found the first endogenous lentivirus in the primate genome. It was in the prosimian lemur.
Lemurs, which live on Madagascar only, which is an interesting question of how
for sure
they got from the mainland.
You have all this paleogeography then going on because you can date, so just like fossils, like bones, you can date those using molecular clock type of approach, very similar to what we talked about earlier with transposons elements. These are type of transposons elements, by the way. Or just by comparing genomes, which species has the insertion in this spot in the genome, which species doesn't. So you can have pretty good, calibration and timing of integration and their age, therefore the age of the virus, minimum age of the virus. And then you can you can really find also clades of viruses that were improved. They were really being co-evolving with their host species for very much longer than what was expected or was possible to see. So this was very exciting. I really like this time.
That began your interest in endogenous retroviruses, which continues to this day.
Yeah.
Right. So you moved to Utah, and then after Utah, you came here. So you like your elements. You like to move around. Right?
Yes. We hop around quite a bit.
Okay.
Horizontally.
So let's Let's go back to the beginnings. And so this is all what you call mobile DNA. In fact, you have a book here.
Yeah.
Called Mobile DNA. This is number 2, but there's a 3, right?
There's a 3.
I think I have the 3.
I like the 2 for personal reasons.
Well,
first we have a chapter in this one. But that's not why.
One of the most interesting chapters, at least in 3, is about Sleeping Beauty. I love Sleeping Beauty because it's one that they woke up by changing some bases, right?
Exactly. Using the same principle I just explained, this divergence, that sort of random as copies accumulate in the genome from this common ancestor. This common ancestor must have been active. You must be looking now at the active element. So if you can take the copies throughout the genome, align them to each other, the mutations that they carry from each other are just discrete mutations often. So by making this sort of consensus sequence along the alignment, you restore the sequence intact, similar in principle to the active element. This is exactly what they did for Sleeping Beauty.
So they made it able to move again.
Yeah.
And what genome was this?
These were from fish genomes.
Fish genomes, right. Okay.
Yeah. Salmonids and things like that. Right. And again, prior to the genome sequences, so they actually painstakingly, isolated copies of these from salmon and so on. Not many copies, I think about 10, I can't remember exactly, but they were lucky enough that with only those 10 copies, and of course they were not too diverged from each other, but they were all like disrupted. Yeah. They were able to kind of erase these mutations and then show that it jumps in human cells, even though it was from salmon. Which, by the way, takes me to another area of interest to us at the time, especially in Texas. And, to this day, I would say, is this ability of some of those elements to go from species to species, hop across.
The mechanisms are universal enough they can do that.
Yeah,
right?
And viruses, of course, are well known to do this. Yes. We know, unfortunately, so.
Indeed, yeah.
On a common basis. But it turns out we and others realized that much simpler transposons, that like these DNA transposons in particular, that just have a transposase, so they don't encode envelope or gag proteins to make viral particles and things like that. They're very simple. Yet somehow they're able to pass from species to species on a regular basis, and we and others have documented jumps like this throughout evolution, including in mammals, of those elements. That's how they got into bats. That's how, we could see very clearly because, you find them in the bat species, but you look at any other bats, you don't see them. You don't see them in any other mammals. Where do they come from? So that was also, it's still a fascinating question. We don't really understand. We think they probably piggyback onto other viruses, in particular large DNA viruses. Such as pox viruses.
They can pick up repeat, because they have big genomes, they can pick up repeated sequences.
Some of these giant viruses. We actually found elements that clearly hop from the host genome onto like giant viruses. Yeah. We have a paper about this many years ago. And so it's plausible that that's the main mechanism. They hop and sort of piggyback onto other elements to pass.
All right, so these mobile elements, they can be DNA or RNA.
Yeah.
But they're always integrated into the genome, correct?
That's the defining feature.
And are they all called transposons also, or not?
Yeah, I tend to lump them all in the same bag. It's a big bag, it's a grab bag. And the boundaries between transposon elements and some viruses in particular is sort of, blurry.
Okay.
We can talk about this, particularly the retroviruses.
And the feature of all is that they can move around, but they don't always move around, right? Because as you said, they're not always moving around.
Yeah.
But they can. And so
they were mobile
they
at
were
some
at
point.
one point, yeah.
They integrated, you're right, that's the defining feature. They integrate in the germline. So that's the other defining feature.
Right, because if you don't integrate in a germline, that's the end. If you go into a skin
end.
cell, it's the
That's
end.
right.
You never
HIV,
right.
immune cells,
That's it, yes.
integrates, cannot be passed on to the next generation through that mechanism.
Okay.
Yeah.
So then the DNA-based ones move by cut and paste. We already defined that, right?
Mm-hmm.
The DNA is cut out and it's pasted somewhere else. So, and how big are these typically?
Those are pretty small. You know, the typical full-length DNA transposon, like the AC element of McClintock, would be about 2-3 kb long.
Okay.
There's basically one gene flanked by inverted repeats, which are the binding site for that transposase that they encode, that one gene they have. That transposase carries out all the transposition reaction on its own. So you can put it in a test tube in vitro, and it does its job, binds to the end of the transposon, cuts as an endonuclease, and cuts the DNA on each side of the element, and catalyze the strand transfer. So that's the catalytic activities of the transposase as well, to another site of the DNA.
I met a guy once who was at a company in Italy who was trying to use these for gene therapy. Does that make sense?
Yeah, with Sleeping Beauty, it's in clinical trials.
Really?
There's multiple clinical trials.
For adding genes?
Sleeping Beauty, piggyback is another element that's being used.
Interesting.
Yeah, for delivery, for gene delivery.
So the transposase, what is that related to? Is it related to a polymerase?
No, so transposase, so now we're going deep origins. So transposase you find in every organisms, basically in bacteria and archaea. You have transposase and you have DNA transposons. What does it relate to? Well, the most common type of transposase is adopt a fold called the RNase H fold, which is also found in RNase H. So we're going back to like RNA world now here, I think, way back. So it's it's this sort of nuclease kind of fold. So they all, presumably these elements, the DNA transposons, the transposase, have very, very deep origins for sure.
And the ones in our genome are only in transposable elements. We don't have any transposases anywhere else, correct?
We have genes that are derived from transposase. And those we have been interested in for many years.
Like what kinds of genes?
Well, so the most spectacular example of a gene domesticated from a transposase that's now important, even critical, essential for human health is RAG1.
Okay.
And RAG2.
Wonderful.
RAG1 is a transposase. It was actually, ever since it was cloned, David Baltimore's lab and others, I think there were several competing groups. It was clear it was a transposase. It was at the DDE, RNase H. Repurposed. And indeed, VDJ recombination. So to explain what this is the process by which, one of the processes by which antibody chains are diversified in a sort of infinite fashion. So that's the backbone of your antibody.
That's a transposase.
The antibody diversification. That is the product of a DNA splicing reaction, a transposition very much reaction, a partial one actually, a modified one, that we owe to the domestication of a transposase about 450 million years ago in the common ancestor of jowled vertebrates. So lampreys don't have VDJ, they don't have antibodies.
Right.
They have their own kind of thing like this, but it's very different. All other vertebrates, fish, lizards, birds, mammals, we all have antibodies. Thank God. Or not God, actually. Thank RAG1.
RAG1, and transposase.
And that's what it does in B cells. And the recombination, the VDJ pasting of these segments in all these different combinations is achieved through this mechanism of so David Schatz now it's clone RAG1, I think, and maybe RAG2 too as well. Still working actually on the origin of RAG1 and RAG2, and actually they found now the closest relative transposon relative of this enzyme, and then been studying in great detail, in structural detail, to understand how do you do the transition from a selfish mobile transposase to this co-opted transposase, which can be very dangerous business, because if there is no reintegration in this case of these circles. There's excision of circles. when they reintegrate, they cause lymphomas. Actually, they do occasionally reintegrate, and they are highly, deleterious. But so there's been modification, obviously, of that system to be able to tame it. And David and others are doing beautiful work trying to understand how does that happen.
So lampreys have transposases, though, right?
Yes, they do.
They just haven't repurposed
they don't
it
have
to RAG1. Okay.
Yeah.
So can you go back? How far? You said RNA world. So you think there were transposases in the RNA world?
Well, in the early DNA world, then.
Okay.
But the fold comes like from that RNase H. Oh, by the way, the fold of transposase is also the fold you see in integrases of retroviruses. So that's going to bring us to the topic of the origin of retroviruses, which is dear to my heart. I'm really interested by this. Clearly, the integrase that you find in retroviruses and also in some retrotransposons, even though they use a copy and paste mechanism, at the end they paste.
Right.
The pasting is essentially the same for retrovirus as it is for Sleeping Beauty or AC maize or for a DNA transposon. So that integrase is really clearly a DD fold, the RNase H fold. So that was captured in these retrotransposons or their ancestors very early on too, because these type of elements, the retroviral-like elements, are essentially widespread in eukaryotes. No, we don't find them in prokaryotes. So somewhere there. So that's a bit later. But anyway, transposase first probably as DNA transposons.
So prokaryotes have DNA transposons, but they do not have retrotransposons.
Yeah, well, they do. They're very distinct from those in eukaryotic genomes. But transposons, DNA transposons, they call insertion sequences, IS elements. In fact.
Right. Okay. So then,
but they have reverse transcriptase and retrotransposable elements, but they're quite different.
Yeah, that's right. Bacteria and archaea both have reverse transcriptase.
Yeah, and group 2 introns, group 1 introns, these are more introns, they have reverse transcriptase, retrons, or oddities.
You mentioned the early DNA world. Was that a precellular world, you think?
Well, that's a big question.
We don't really know, right?
No.
Nobody knows. So there were transposases present, so probably there were transposons, right?
I tend to think that as soon as you have, replicative mechanism of nucleic acids, you're gonna have sort of self-replicating entities that gonna cheat the system at some point too. As you become cells, you're gonna have some that take advantage of replicate faster than the cells and sort of become selfish elements. So whether this is RNA or DNA based.
All right, so the other kinds of mobile elements are RNA based, because as you said, the DNA is copied to RNA, which is then reverse transcribed to DNA and then integrates somewhere else.
Yeah.
And so those are called retrotransposons.
Retrotransposons.
Right? Because their activity depends on reverse transcriptase, correct?
Yep, correct.
And those are also ancient, right?
Presumably very ancient. Those may go way back to the RNA world.
Right.
In some form, Reverse transcriptase presumably was the enzyme that must have copied RNA to DNA. Right?
To make the DNA world, right?
Yeah.
Yes, that just arose probably from an RNA polymerase and
I would
randomly,
think so.
And here we have us as a result. So, and these encode reverse transcriptase.
At least. And then
integrase as well?
So that depends on the flavor of elements that you have. And so in eukaryotes, at least, there is two major types of retrotransposons. Those that have the long terminal repeats, LTR retrotransposons, they resemble retroviruses very much.
Those are products of retroviral integration, right?
Well, we'll talk about this. We'll get back to this in a minute. But these are the LTR, so-called LTR, long terminal repeat retrotransposons. So their genes, their coding genes, including the reverse transcriptase, which is in the pole open ring frame, is playing by these long repeats. And these are essential for the way they replicate. And the characteristic of those elements is to do reverse transcription in the cytoplasm of cells, much like retroviruses. And then the other major type of retrotransposons are the non-LTR retrotransposons. So those that don't have LTRs. And the characteristic most one of the most best studied elements of this type is called line 1 or L1 in the human. It's the only fully active element in the human genome. Line 1, it's a non-LTR retrotransposon, also known as a line element. Those do reverse transcription in the nucleus through a very different mechanism than retroviruses that's called target prime reverse transcription, when they initiate reverse transcription straight onto the DNA, so therefore in the nucleus, by nicking one or two strands of DNA, which serves as a primer, because reverse transcription, the trick is like you need to prime it. You need a 3' OH.
Right.
Those have an endonuclease that they encode, so they encode also an endonuclease activity that nicks the DNA, and that creates the 3' OH. And then their RNA anneals to these through their poly-A tail, because they cleave at like a site that has a TTT, right? So cleave like this, and then that initiates reverse transcription into the DNA. So it's target prime reverse transcription. That was discovered in the silkworm by Tom Agbush's group in the 90s, and it was really kind of bizarre and unexpected. Everyone thought that probably they were replicating much like retroviruses, but we didn't understand the priming mechanism. So LGR retrotransposons and retroviruses do reverse transcription in a viral particle,
right?
Typically, and use tRNAs typically as primers for that reaction to provide that free prime OH.
Cytoplasmic, right?
Yeah. So really very different mechanisms.
Yeah.
Yeah.
So reverse transcriptase in the nucleus must have a nuclear localization signal, but not retroviral RTs do not have a NLS, correct?
I don't think so.
Yeah, I don't think they do. So these are very different. These are cellular RTs that.
Yeah, and it's a ribonuclear particle RNP complex that assembles in the cytosol that includes the retro element RNA bound to at least that one protein that has reverse transcriptase and endonuclease activity. It's called OF2P, it's called for line 1. There's also an OF1 that kind of functions a little bit like the GAG. It's like a chaperone, but it doesn't make a particle, but it coats the RNA. So it's kind of you can see the ancestor of retroviruses there, because it's kind of similar. But that RNP then must traffic into the nucleus. Actually, the trafficking, how it happens, not very well understood in any system, I think. Yeah.
So the retrotransposons, they're lacking envelope. Do they have GAG? I know they have POL.
Yeah.
Do they have GAG and POL, but no envelope?
So the LGR retrotransposons have the same exact genetic, nearly exact organization as retroviruses. So the GAG, POL, but they do not typically have envelope. And there are exceptions.
So the endogenous retroviruses have POL. So
they have envelope too. The endogenous retroviruses.
Yes, they have envelope. That's
some of them do.
Yes.
Because that's why we know they come from retroviruses, actually. And they're not LGR retrotransposons. Also, you can take the reverse transcriptase and they group with retroviruses and not with the other LTR retrotransposons.
So, I have always thought that the retrotransposons were the precursors of retroviruses, and all that was needed was to capture an envelope somehow, which I don't know, maybe Eugene Koonin may think comes from the cell, right? Many years ago, they pulled, and it made a particle, and that made a virus. Is that still what we think?
Yeah, so that's a really interesting question, and I think we have a pretty good handle on that one, on the origin of retroviruses. I think this may actually be, correct me if this is wrong, but Eugene would know that, but that may be the only group of viruses for which we kind of understand where they come from, how they get together, how they got pieced together.
He would say there are others now too.
Yeah,
you can find capsid signatures in cellular.
Yeah. There's signatures for sure. That is really interesting. I love this topic of origin of viruses. But so retroviruses is quite. Let me. What do I call retroviruses? So typically the Retroviridae. So that's the family of retroviruses that you find in vertebrates. And so first of all, they're unique to vertebrates. Okay, so if you make a phylogeny with reverse transcriptase from all the known retroviruses of vertebrates, they come together. They presumably have a common ancestor. Probably then have acquired, if they were born by acquiring an envelope, this must have happened only once, although it's not entirely clear, but they come together. So they make a group. So this is what I call retroviruses, right? This group is sister in the phylogeny of reverse transcriptase to the LTR retrotransposons. There's two different, several major classes. I'm not going to get into this. And these LTR retrotransposons don't have an envelope. They have everything else: GAG, POL, integrase, reverse transcriptase, RNase H. Those are very widespread in eukaryotes. These are probably maybe the most successful transpose elements on the planet, because you find them in plants, you find them in protists, you find them in plenty in insects and invertebrates, plenty in vertebrates that also have retroviruses. So like fish, they have these kind of elements, but they don't have an envelope typically. So the preferred scenario, then the most parsimonious scenario, is that these type of elements, the LTR retrotransposons, are ancient. They've been around for a while, and somehow one acquired an envelope. Yes, the origin of the envelope or the vertebrate, the retroviridae, is mysterious. We don't know where it comes from. It has no homology that I know to anything. They are also very different from each other. That's why we cannot rule out multiple capture of envelope. In the common ancestor, probably of vertebrates, about maybe 500 million years ago, the acquisition of that envelope facilitated the ability of these elements to escape the germline and become infectious. Now be able to also affect somatic cells, go from cell to cell, from individual to individual, and maybe also species to species. So I think we have a good handle on this, because there is actually evolution, as has repeated itself. Yeah. And there is a beautiful story in Drosophila of retrotransposon, a typical LTR retrotransposon that has acquired an envelope completely independently from the retroviruses of vertebrates. Mm-hmm. An element that used to be called gypsy. Right. We don't like to call it gypsy anymore. Yeah. There's a lot of talking about this these days. We were just talking about this with the lab the other day. We prefer to rename it now MDG4, which is the way the Russian group that actually independently identified these elements, Georgiev's group, called it originally. So we go back to this name. Anyway, MDG4, gypsy elements in flies, those have an envelope.
Right.
And where the envelope comes from? It's actually pretty clear. It comes from a baculovirus. So a completely different virus, of course, than a retrovirus, a DNA virus, because it has direct sequence homology to the envelope you see in baculoviridae. So presumably,
which can affect the Drosophila, is that correct?
Yeah, insect viruses. Yeah.
And did baculo get it from a cell, from another insect maybe? Who knows?
Who knows? Origin of envelopes, very interesting topic. But
Let me
just pretty clear.
Let me just check something first.
It's pretty clear that the gypsy envelope, MDG4 envelope.
Can you move over? We've drifted, so I want you to stay closer. I didn't know that.
So that's basically it's a replay, probably, on what happened in vertebrate, or during early vertebrate evolution. And by the way, Harmeet Malik, Harmeet,
yes.
Published a paper during his, let me try to get this right, his postdoc with Steve Henikoff, called, the title of the paper is Poised for Contagion. Because he basically, by doing some sequence mining of LTR retrotransposons, found that multiple cases of LTR retrotransposons in diverse organisms, in worms, not just in fruit flies, had acquired genes, additional genes in the same exact position as the envelope, with transmembrane domain that look basically very much like envelope, but had also matched to other types of viruses. So that's a remarkable paper, actually, showing that this capture of envelope from viruses by retrotransposon has happened multiple times in evolution, and presumably enabled these elements to become infectious. In the gypsy and DG4 elements in Drosophila has been shown to be infectious, passed from cell to cell. The other ones, we don't know. No one studies them.
Right. Okay.
But they are composite chimeric elements with envelopes. So presumably, retroviruses emerge repeatedly during animal evolution. And even in plants, there's actually elements in plants.
Sorry.
In plants too, there are elements like typical LTR retrotransposons that have additional open reading frames that look like envelopes.
So these retrotransposons, whether they be RNA, LTR or not, or endogenous. This is not the majority of transposons in mammalian cells anyway, right? Or most cells. They're a minority, correct?
Yeah, they're a minority. So the endogenous retroviruses. So the mystery, I'm not going to get into things that are too esoteric here, but one question that keeps me up at night is like the traditional LTR retrotransposons. I talked about these MDG4, gypsy elements type. I told you it's the most successful elements. There's 80% of the maize genome is made of this type of elements. They're very common. They're completely absent in the human genome. So I don't know why. And they're actually absent from all mammalian genomes. So they disappeared. But they got replaced by endogenous retroviruses. And now we do the reverse movement. Instead of escaping by acquiring an envelope, now we get trapped in the germline by losing an envelope.
Yeah, right.
So the reverse movement happened. And those elements now have transformed into retrotransposons. They clearly derive from exogenous retroviruses. You can sometimes trace them to things that are still infectious, like MLV, murine leukemia virus in mouse, as exogenous and endogenous elements. Or in the koala, there's an infection, an ongoing infection, an invasion of the genome. So they found their way in the germline. And the process that has been described by several groups seemed to be like they tend to lose their envelope, but they continue to do retrotransposition in the germline, germ lineage. And they spread in the genome like retrotransposon, like transposon elements. So about 8% of the human genome is composed of these type of elements we call human endogenous retroviruses. 8%. So it's 8%, but as opposed to about 30% of the genome made of retrotransposons of the non-LTR types.
Got it. Okay. So I'm trying to, I know that people have found repurposed elements from endogenous retroviruses, like famously syncytian, right? And then Ed Trong and you and Nels, the promoters used for interferon responsive genes, right?
Yep.
And there must be many others as well. But then there's other parts of these herbs that are not apparently used. So why are they still there? Mm-hmm. There's always a question that is fascinating. Is there enough room in the genome that it doesn't matter, or is there some benefit?
Yeah, why accumulate so much potential junk? And again, I do believe that most of this DNA is not functional, is not doing anything. It's accumulated. I mean, there is all the signatures that we expect of neutrally evolving sequences on those elements. Of course, we look for the exceptions to this. Mm-hmm. How many exceptions there are, meaning exceptions, elements that were sort of co-opted for cellular functions that are actually now important. So now we cannot remove those because they are under selection, like any genes. How many of those? We don't know. The list is growing, but certainly not the 8% of the endogenous retroviruses that we have in our genome. So why then are they there? Well, they are there because they can.
They can be. That's the answer.
Can be. It's a very simple answer, but in a sense that it's known that in organisms with very small effective population size, so now we're going into like population genetics, the genetic drift as opposed to natural selection, genetic drift, so the fixation of alleles or mutations by chance, if you want, is very strong. So you will get fixation of even like slightly deleterious things that you don't do anything good for you, but they actually may even be a little bit bad. They would still get rich fixation by chance alone. So that's a genetic drift. In organisms with small effective population size, such as vertebrates, including humans, its genetic is very strong. So you can't stop it. So they will get fixation. So you get this genome bloated with stuff that would be removed if the organisms had larger effective population size. So in fruit flies, you would see much less of this DNA accumulate. And indeed, they have in bacteria or in yeast, in between yeast, it would be sort of in between fruit flies and bacteria. In bacteria, enormous effective population size, which means that natural selection is very strong. Anything that's bad is going to be eliminated very quickly. They would not accumulate typically large amount of non-functional DNA, including transposable elements. They do have some, but they're all very recent. You don't see the old stuff hang around.
Now, where do humans fit? Are they a small population size?
Very small effective population size.
So we get rid of these.
These are effective.
We accumulate them.
Yeah.
Right?
Just to make sure people understand what I mean by this is like effective population size is the size of the population that can reproduce with each other. Yeah. So it's on the order of 10,000, I think, in humans. It's actually very small. It's not the global size of the population, obviously. So it's, the individuals that can mate, basically. So that's your effective population size. So humans are really on this end, really the end of the spectrum when it comes to the efficiency of natural selection, not very, very efficient, and drift very strong. And many other vertebrates. Probably that's also why salamanders, have genomes 100 times bigger than the human genome. Mm-hmm. Almost completely made of transposon elements.
And say corn as
Mostly
well,
these retrotransposons, corn
as well. And so
the
population of corn is small as well, correct?
This is a theory, yeah? Okay. This is population genetics, so we're going into theoretical things. Now, is there really good? This, by the way, this is a model that was, I think, first proposed by Mike Lynch, population geneticist. It holds, it makes sense. Now, when you start looking at differences, it cannot explain very quick variation in genome size between closely related species. Right. So between corn and like sorghum, which is another grass, that's a genome that's much smaller, 3 or 4 times. Doesn't have a very different effective population size. So clearly that cannot explain all the variation in genome size. Right. Okay, but it may explain the big patterns.
Yeah.
The bacteria, the fruit fly, the yeast.
Okay, all right. So in some cases, though, these elements can cause harm, right?
Yeah, for sure.
I think you said
different.
Yeah, you said 120 independent insertions have been associated with human disease. So this can be a DNA or an RNA.
Those are the few elements that can jump around in our genome.
So how many are active in our genome? Do we know?
So remarkably few. I would say this is still why. For example, you can mouse, you have a lot more active elements going on. You look in fruit flies or in maize, there's a lot more going on. Humans don't have a lot of active transposition compared to these species, but we still do. So to answer your question, so let's just look at the landscape of the human genome. So we have millions, something maybe 4 or 5 million elements like this in our genome. But that's interesting, put that in perspective to the 20,000 genes, right? We have
protein
millions.
Yes, millions.
And many are actually
and they're there
coding.
because they can be, right?
Yes.
And many of them have undergone drift so that the transposase is not functional anymore, right? Okay.
Yeah, exactly. So they are pseudogenes in a way.
All right.
Initially, they may have been counted in the human gene count, and then we would end up with 500,000 genes or maybe more. So, so there's all these elements. So of those, there's one element. We talked about Aloe elements. So Aloe is an interesting one because it doesn't encode anything. It's a parasite of a parasite. It's an element that parasitized the line 1 element that we talked about earlier, a non-LTR retrotransposon. Turns out Aloe, as its completely independent origin from line 1, it comes from a functional RNA called the 7SL RNA, which is a translation involving translation. And somehow it's being recognized by the machinery of the retrotransposition machinery of line 1 in trans. So it's mobilized very efficiently.
By line 1.
By line 1. Line 1 probably doesn't like that because it just sucks its resources, So it's probably a parasite of line 1, but it's a very successful one. It turns out there's more Aloes than line 1 in the human genome. So we have 1 million plus Aloes, and we have about 600,000 line 1 elements. So a lot of these line 1 elements. Line 1 is the only fully, what we call autonomous transposable element in the human genome, fully active. Of the 600,000 line 1 like things in the human genome, about 100 are capable per individual, per haploid human genome. About 100 are capable of moving around. That's about the estimate at the moment. These, interestingly, may be different between you and me, because those are very recently active. So they've jumped recently. So they're still polymorphic in the human, insertionally polymorphic in the human population. So that's an interesting to think about, because you and I, Vincent, we may have different rate of transposition going on because we have different subset of active line 1 elements. But anyway, it's kind of astounding that such a very, very small sliver of those elements are, and it's a good thing probably, right? Because indeed, when they jump, they do cause disease and they cause problems.
You say that they're active. So can you give me a time frame? So, right, and it has to be in germ cells because it doesn't matter in somatic cells. We don't care.
For the elements, but they still are active in somatic cells, and it's a paradox at the moment.
And they may have some transient phenotypic effect, I guess, right?
Possible, yes. We're really actively studying this question, actually.
But in a germ cell, what's the time scale of activity? Is it months, years, or decades?
So how frequently do they jump in humans?
Like every time DNA reproduces, maybe?
No, so in humans, the frequency of line 1 retrotransposition or Aloe is about the same too. Aloe still jump as well, right? In fact, they jump more often than line 1. Together, it will be about one new jump, so a new element in the germline, so passed on to the generation from parents that didn't have this insertion. So the insertion will happen in the egg or the sperm or their lineages. Right. About one new jump like this per 20 human birth. So not too frequent.
Not very frequent.
Not for every new baby out there is a new de novo insertion in the germline. Right. In somatic cells, it's much harder to trace. But interestingly, if you look in mouse you have a lot more going on. And I don't know what the number would be in mouse, but it may be one every birth.
Mice are interesting because they also make virus particles from endogenous retroviruses.
Yes, they also have active...
And they infect other mice, right, horizontally.
Yes. Yeah.
And I don't know if that's a function of time. Eventually that will stop, or it's just some unique aspect of the mouse population.
Yeah, that's right. Mouse are also different in the sense that they have active endogenous retroviruses that are capable of retrotransposition and also infection, and or both.
And it's possible that they may sustain insertional mutations, but we don't know because we don't look at wild mice very much, right?
Not many people look at wild mice for retrotransposition, I think.
And so what about laboratory mice? Do they have similar rates of retrotransposition and transposition?
Higher than humans.
But compared to wild mice, similar?
Oh, I don't think, we don't work on mouse, so
no, okay,
I don't think it's been looked at in wild mice population. Not with the same level of precision as in the lab mice. Okay. The lab mice, there's some really nice studies from Jeff Faulkner and others, quantifying how much transposition there is per generation per new mice. There's definitely a lot more than humans.
So how would it go that a mutation would occur if an element moves into a gene and say disrupts it? If that were in a germ cell, then that would have to end up in an offspring, and then depending on whether it's dominant or not as a heterozygous, right? That would matter. But if it's not, then you would never see it.
Yeah. So exactly, and it gives you even more appreciation for how much was happening at the level of the population to end up with so many elements. So that's another thing, population genetics, if these elements get fixed mostly by drift, which they must be in humans, that's probably very clear, then we see those that are fixed, meaning they are shared between us, so the 99% of those elements in our genome are really only the tip of the iceberg of the elements that were segregating in the population, really. Because the likelihood of fixation of these new insertion, population genetics tells us is 1 divided by 2 times effective population size. So 1 in 20,000 chance, by chance alone. Right? So you see, that's why it's more likely than if you have 100,000. There would be 1 in 200,000. But still, it's extremely rare.
Okay.
Yeah.
Now, so the fact is that some genomes have so many of these elements because they can be there and they're not detrimental, and they can be inactive and it doesn't matter. So, and some, as you said before, some organisms have very large genomes because they have small effective population sizes and they don't get weeded out.
And maybe other mechanisms by which these elements can, so the control, we haven't talked much about the control mechanisms, but we know of multiple mechanisms. Some appear to be specialized in repressing transposable element activity in the germline. Those mechanisms are also highly variable between species. So it's also possible that it shapes how much, elements you have in the genome. It's not just about population genetics, it's also like host factors, essentially.
Yeah. So in a bigger sense, so these are, so someone, my colleague Dixon Despommier, who does TWiV with me, he says, successful systems attract parasites. So that's what these are. These are all parasites that are piggybacking onto genomes that are replicating, and there's obviously no intent, but they're just on a molecular level things that can arise when you have a system that reproduces, and so small parasites arise that take advantage, right?
Yeah, I think that's the way I tend to think about it too. However, just like any other parasites, they may evolve into very interesting relationship with their host.
Right.
And this is one of the things that we're really excited about in the lab at the moment, is to try to explore the idea that sometimes they may not be just conflicting relationships, but more long-term, more like mutual relationships. Because indeed, as opposed to viruses that infect somatic cells that are sort of much more like on an aggressive evolutionary line, these elements that we have in our germline have to kind of co-evolve with their host. They can be overly active, for instance, and we know of many mechanisms that repress, self-mechanisms that repress, where the elements are sort of suboptimal, right? So we talked about Sleeping Beauty earlier. As a very interestingly, Sleeping Beauty, was found to be put together as the ancestral element, put in human cells. It jumps at some frequency, not a lot, but it was very easy to turn Sleeping Beauty into hyperactive transposase. Any transposase you can take, you can basically, by mutagenesis, find hyperactive versions by changing one or two amino acids. So it tells you something that during evolution, there is some pressure to minimize to the rate of transposition, and these elements are sort of selected to be suboptimal. So anyway, that's just an example. Like, this is probably you won't see this in a virus, or maybe less likely. I think these germline parasites actually are more likely to evolve mutualistic relationship with their host than other parasites. So I think that's one thing that has not been really explored very much. The idea that in fact there may be a give and take, right? So maybe sometimes they do something good that favor their own propagation as well. So we are really testing this.
You don't know what that might be yet, right?
So it's in progress, research in progress. But we basically, so the beauty now is that we have the tools to test this. Honestly, this was an idea that was brought up by one of my graduate students, Sylvia. When she joined the lab, she was like, this is what I want to show. I want to just test this idea that maybe they are actually beneficial, these elements, as active elements. So not these old, ancient, co-opted pieces here and there that we've been studying, which are kind of cool examples, but they're no longer transposable. They're no longer active, right? No, she really wanted to look at elements that are just behave as a transposable element. They jump. They're active in a population, but show that now they do something. So we can do the genetic manipulative experiment of removing them or depleting them, and see if you have a phenotype, right?
Yeah, I was going to ask
exactly what she's doing, and she's using the zebrafish.
Because you can deplete them from the zebrafish?
Yeah, and also we were looking specifically for embryonic phenotypes, because we were finding elements that were active in a very early embryo, and zebrafish is very tractable as a system for embryonic development, and looking for phenotypes. And we're starting to see things that seem to be consistent with the idea that some of those active, these are LTR retrotransposons, by the way, retroviral-like elements. They
not herbs, obviously, because it's zebrafish.
They are herbs, actually.
Yeah,
they are, but they are like fish-like retroviruses. In fact, these are, and so I don't want to get into too much detail because it's completely unpublished work, but it will be hopefully published. But using. So now we have the tools, right? I was saying, like, we can use CRISPR, we can use morpholigos, antisense oligos, pretty sophisticated techniques to deplete those elements. And that's what she did. And she's seeing things that are consistent with the idea that you need a certain amount of those elements. Mechanistically, we are thinking it has to do with the GAG, the viral particles, and basic processes of development that involve cell-cell communication and signaling that these elements kind of...
ARC.
Yeah, right. That was inspired by ARC, in fact, all this work.
Okay.
We think that these elements modulate, they have the ability to encroach on development. And in a way that maybe not somewhat benign at first, but as they increase into copy numbers, the dose of these proteins starts to modulate development. And we think the way I think about it, at least, and I think we're starting to find evidence for this, is that they create what I call an addiction. So this is what we call the addiction model. Now the host become addicted to certain dose of these products because you have had to adjust. Again, this is owing to their coexistence for many years. For many years, you have to adjust.
Exactly.
Now you can no longer remove them.
Yeah, exactly right. Makes perfect sense.
Yeah, so it's testable.
So you're just removing one kind of mobile element,
very
not
specific
everything.
kind.
Not everything. That's good, because to remove everything would be very difficult.
It would be a mess.
Yeah. So then,
very difficult
for people who don't know. ARC is a gag-like particle which is thought to be involved in CNS communication. With Jason Shepard, when you were in Utah,
we work with Jason. We're still working with Jason. Actually.
Yeah, Jason is on another podcast.
Yeah.
We do This Week in Neuroscience.
Oh, yeah.
And he's talked about that as well. Yeah, so that I was going to ask you, if you remove the mobile elements, what happens? So you're doing that with a specific kind. So
we're doing that.
That's interesting.
And it was very much inspired by Jason's work on ARC, because we think that in fact what ARC is doing now, ARC is clearly a co-opted. It is no longer a retrotransposon or retrovirus. It's derived from a LTR retrotransposon ARC. And but what it's doing as terms of packaging and on RNA trafficking potentially at the synapse, we think in fact these are not new activities of those elements. Again, this was borrowed from things that these elements do elsewhere as well, not just in the brain, in particular where they like to be active in the germline, in the early embryo. This is the battleground really. And these activities actually have an impact as the elements are active.
Let me just qualify one thing, or understand one thing. So you've said that many of these elements are inactive, right? Mm-hmm. Yet, if you take them out, there's a problem. So does that mean the minority that are active are the ones that are contributing? Yes?
Yeah, well, also I need to say that in human, it's very skewed for inactive elements, but the zebrafish is a lot
active.
Going on.
Oh, okay. Okay, so that makes sense there. So it may not be this case in humans. We can't do the experiment, but...
Well, so there is some mounting evidence that the line 1 element that I mentioned, which is still active in humans, at least in the mouse, I should say the evidence comes from mouse work. There is several studies that have been published that suggest that the line 1 RNA itself may have modulatory activity on early development. And it's been also tested with depleting that RNA doing experimental work, suggestive that in fact they've proposed this idea of cooperation or partnership between line 1 and the host for development. So this idea has been flowing around too for line 1. It needs to be tested. I mean, we chose to work on the zebrafish because it's more tractable than mammalian system, we think, for this, but I think there are people working on this.
Well, when you publish that, we'll have you and your student come and talk about it. Line RT is cytoplasmic, right?
The line 1, they need to go in. So there is a bit of controversy at the moment. I'll tell you why. But the canonical retrotransposition pathway of those elements is to do this target prime reverse transcription in the nucleus. However, the RT is expressed obviously in the cytosol of cells, needs to be trafficked to the nucleus, but there is evidence, growing evidence, and this is an area that's very active at the moment, you may have heard about it, for the presence of DNA, line 1 derived cDNA, so product of reverse transcription, presumably, in the cytosol of cells, where it would not expect to see it. Yeah. And this is attracting a lot of attention at the moment because it's sensed by the viral sensor like C-gas and these DNA sensors that normally sense bacteria or other viral DNA also can sense this cDNA derived from line 1. And this, if you have too much of this layer, in particular during aging, you have chronic inflammation essentially. And so it's basically, it looks like line 1 derived cDNA in the cytosol accumulate in certain condition during aging or in other, maybe in neurodegenerative disease, such as ALS, it's been seen as well, that contribute in part, to sort of chronic inflammation that's characteristic of aging, characteristic of many, neurodegenerative disease, autoimmune disease as well. So that's a really active area at the moment. The question is, where is line 1 CNA doing in the cytosol to begin with? Shouldn't be there. It's a mystery at the moment. It's possible that you have some level of RT activity of line 1 in the cytosol of cells, sort of aberrant in a way. It's plausible. Or there is trafficking of that DNA on the nucleus, leaking out of the nucleus. That's possible as well. But that's an interesting area because now it's another way by which transposon elements seem to be capable of affecting physiology and potentially development, biology of organisms, not by their transposition activity again, but by their products.
Right. So a couple of groups took L1 expressing cells and infected them with SARS-CoV-2, and then they say it's reverse transcribed and integrated. I mean, what's the significance of that? I find it hard to explain.
First, with regard to this particular work, it was not entirely convincing that there was integration. Are you talking about the integration of SARS-CoV-2 DNA that would be reverse transcribed, right?
By line 1 RT, right?
Right. First, there's two competing reports. And one that argues there is such a thing happening in human cells in a fairly artificial system, and another one that tried to replicate this experiment and found zero or undetectable. So up in the air about what's the frequency. And I would say mechanistically it's totally plausible. We know the line 1 machinery can grab onto any RNAs.
So it presumably does it with cellular mRNAs too.
It does it with cellular RNAs. There are retrogenes.
Yeah.
You've heard about those from nails, right?
Processed pseudogenes.
Extremely abundant. Yeah. Thousands of genes like this have been clearly grabbed by the line. And we know it's line 1 because it's all the hallmark of the line 1 mediated reverse transcription.
So presumably any RNA virus could do the same thing.
And it's been reported before for virus, which is a much more obscure RNA virus.
Yes.
And the data was quite convincing that virus established these sort of permanent infections of cells. After a month looking at cells, when you infect them with virus, you can find integrants for virus. So it's not totally implausible that this would happen. Now, the impact, the clinical relevance or pathogenic relevance, all depends on the level of, on the rate by which this would happen. And the data so far would indicate that if it happened, it's extremely infrequent. And where it happens would be also very important, of course, in terms of physiology, right? Which cells? Is this an integration in one cell that's non-dividing, like a muscle cell?
Right.
It's the dead end.
Dead end, yeah. Okay. All right. I have to end this. I can go on for many hours, but but I have to move on. So that's.
And I didn't tell you, but we have an upcoming story. I think you enjoy it. We can chat again at this point when it comes out.
For sure.
It's accepted in principle at Science, and there's a new study looking at envelopes derived from human endogenous retrovirus that block against exogenous retrovirus, so they have antiviral activities.
Antiviral activities.
So that's pretty cool.
Oh, cool.
So we're excited about that one. I'll tell you that. Yeah, I'll tell you about that one when it comes out.
When it comes out, we'll have you back to talk. Yeah. All right. This is a special TWiV in Ithaca at Cornell. You can find the show notes at microbe.tv/TWiV. You can send questions and comments to TWiV at microbe.tv. And if you enjoy our work, please consider supporting us. Go to microbe.tv/contribute. My guest today from Cornell University, Cedric Feschotte. Thank you so much.
Thank you, Vincent,
for talking with me. I appreciate it. Fascinating. We could keep on talking as well. Yeah. I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Virology, the American Society for Microbiology for their support of TWiV, Jolene for the timestamps, and Ronald Jenkins for the music. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.